Okay, so you guys know the drill. We're taking notes on proteins, and you need to have your graphic organizer out in front of you. Proteins are going to be a lot like carbohydrates. You're going to have a monomer that's repeated into longer units to make polymers. So very similar pattern to what you saw with the carbohydrates. Proteins have one additional element in them past what the carbohydrates and lipids had. They have hydrogen, oxygen, carbon, and nitrogen. We call the monomers amino acids, and we'll talk about why we call them amino acids in just a second. And when the amino acids link together, when the monomers link together in a condensation reaction, we call the bond between the two a peptide bond. When we have lots of amino acids linking together, we call that chain of protein. We give it the name of a peptide bond, this special bond. Make sure you jot that down, just because there's so many proteins in our body and they have so many special jobs. So it is still just a condensation reaction, but we give it a special name. Basic structure of a protein looks something like this. This is one amino acid. And I told you I would explain why it's called an amino acid. Proteins have a central carbon. You can see that right here, one carbon in the middle. And like we know from earlier in the notes, carbons like to bond 4 times. Proteins bond the same way over and over again. On one of the bonding sites, we simply get a hydrogen. On another bonding site, we get something called an amino group, a nitrogen bonded to 2 hydrogens. On the other bonding site, we get something called a carboxylic acid group, a carbon double bonded to an oxygen and single bonded to another oxygen and a hydrogen. This is where we get the name amino acid. It's named after the groups that are bonded around that central carbon. The 4th bonding site is called the R. R stands for radical group. Make sure you write that down. R stands for radical group. And different things can go here. It's kind of like a variable position. And we'll talk a little bit about what can go in that position. In this other picture, you actually see 2. Sorry about that. You actually see 2 amino acids bonded together. Here's a central carbon. Here's another central carbon. And right here, this is where my condensation reaction or my peptide bond is forming. A water is going to be lost as they're put together. So this would be the beginning of a protein. We could call this a dipeptide or a 2-unit protein. So the dimer is called a dipeptide. If I continued linking through condensation reactions, I would have a polypeptide. Again, I told you guys this was very similar to carbohydrates. Carbohydrates have monosaccharides, disaccharides, and polysaccharides. Here we have amino acids, dipeptides, and polypeptides. There's 2 different forms that proteins can take. Proteins can be fibrous or globular. So we're talking here about these long polypeptide chains. Proteins that are fibrous would be things that are like long, thin strands built of thousands and thousands of proteins bonded together. So things like your hair. Your hair is just one long chain of amino acids linked together into one long polypeptide. Or spider webs. These are just chains of proteins. Those are considered to be fibrous. So think of the word fiber, like fiber in a rope or a string. Globular proteins really are just like little blobby globs in cells. And I know that's not a technical term, a globby blob, but they're molecular little blobs, small protein molecules that float around in your body and have different jobs. The most common protein, globular protein, is hemoglobin, and it's a little blob that floats around in your bloodstream and carries oxygen around. So in general, the fibrous proteins are very large and the globular proteins are very small. The 2 main functions that proteins have in your body are to build up your body tissues. You probably know that your muscle is made of a lot of protein. So if you're trying to build your muscle, you're going to have a diet rich in protein. And they also form enzymes. And we'll talk later in the notes about the roles of enzymes in your body and why they're so important. Here's a picture of one of those globular, globby little amino proteins, hemoglobin. So you can see this is on the molecular level. This right here, this is one iron atom. So if you're talking about something that's very small, if you can see an atom, but the different colored portions of the molecule, those are just proteins in a chain, a short chain, forming a globular protein. And oxygen would sit right here. So this molecule carries oxygen through your body. Now we do need to talk a little bit more about that R group. So the R groups, I told you that there can be different things in this R position. So when you look at an amino acid, you'll always see that central carbon. There will always be a hydrogen bonded, and there will always be an amino group and a carboxyl group on every single one. But that R group is kind of like a variable position, and as you can see from the picture here, you can attach different things to it that will give the amino acids different properties. So this is a little different than carbohydrates, because in carbohydrates you have the same monomer over and over and over and over and over again. So when you have your monosaccharides linking to form polysaccharides, your polysaccharides are very similar to each other. You can only get so many types of carbohydrates. But in proteins, there are actually 20 different things that I can put in at this functional group. So I have 20 different monomers because I have 20 different R groups or functional groups. And so I can get proteins with lots of different functions and lots of different characteristics. So that R group, again, we call it a functional group. It's just a little cluster of atoms. And I put some examples here just so you're aware of them. When you're hearing these words, you'll know that they're functional groups. Hydroxyl groups, they have an O and an H. It would look something like this, just having an oxygen and a hydrogen. Phosphate groups we'll see later in our nucleic acids. Carboxyl groups you have down here already. Carboxyl group is a C double bonded to an O, bonded to an O and an H. So it's just these groups with different properties. Again, it gives you a much broader variation of types of amino acids. Okay, here's a list of some functional groups. You don't need to memorize this list. This is something that if you took like an AP biology class or a college level biology class that you'd start to talk about. Even some college chemistry classes, especially organic chemistry classes, will talk about these because the properties of the functional group will tell you a little bit about the molecule. So I don't want you to worry about memorizing the different formats that a hydroxyl group is a hydrogen and an oxygen, and an amino group is a nitrogen and 2 hydrogen atoms. But I just want you to be aware of all these different molecules that can be attached at that functional group and give the amino acid unique properties. Okay, and here's just another view so you can visually see. So here's that hydroxyl group, just the oxygen and the hydrogen. Here's the phosphate group added on. Here's a methyl group. And again, we are not going to get into the properties, but scientists can study these and learn more about the molecule depending on which functional groups are attached. The test for protein, really simple. It's going to mimic the same set of steps you took for the iodine test. I want you to use a spot plate for this protein test and use the testing solution called biuret. Biuret starts off as a very, very pale blue, like you can see here in the picture, but when it comes into contact with a protein, it turns a lavender or a light purple. So I want you to add equal amounts of whatever solution you're testing to the same amount of the biuret solution and see if it turns the purple color. If it turns the purple color, there's protein present. If it doesn't turn the purple color, there's no protein present. Run this on the spot plate. Again, visualize how much liquid you think you'll need to add, probably between 2 and 5 drops of each of the solutions.